Christian Hansen has been improving food and health for more than 140 years, and to this day, we produce ingredients consumed daily by more than 1 billion people worldwide. We work continuously to develop the products of tomorrow, enabling farmers to produce the high-quality, sustainable, and safe food that global consumers demand. All this is accomplished from our strong platforms in bioscience technologies, combined with extensive research and in close dialogue with our customers and business partners. This is why we call our approach science-based, research-proven.
Okay, good afternoon, everyone, and welcome to Concurrent Session D, which is What's New in Research at Iowa State. My name's Jamie Studer, and I'll be moderating this along with Blythe Schultz, and we're both graduate students here at Iowa State in Dr. Jason Ross's group. So it's my pleasure to introduce Dr. Laura Greiner, who is our first speaker. And she's an assistant professor at Iowa State University, and upon completing her PhD in animal nutrition, she spent years conducting research within a production company in the United States. She currently has 4 graduate students, and she teaches multiple undergraduate classes focused on swine production and assists producers around the state of Iowa. Her current lab focuses have been understanding compensatory gain in wean-to-finish barns and discovering new opportunities to improve sow longevity and piglet survivability.
Thanks, Jamie. Well, I'm the first one after lunch, so hopefully this will be entertaining enough to keep you all awake. It was a good lunch, so I do want to thank TechMix for providing that very good lunch for us. Well, I think we'll go ahead and get started. As the title suggests, we're going to talk today basically from weaning to lactation, and it's a very long process, and I understand that. But what we're going to talk about really focuses on gilt development and sow longevity. So we are doing some work in our lab where we're working on still some follow-up on compensatory gain during the COVID and corn diets, but that work's not ready yet. Chloe will be finishing that up and sharing that very, very soon. So today we're going to talk about some of the work that we've either recently published through abstracts at Midwest or work that we're getting ready to publish out into the industry. So as Jamie alluded to, I am currently an assistant professor here at Iowa State. I've been here now for about 2.5 years. So as it's mentioned as well, my focus is mainly the undergraduates, so about 50% of my time I spend introducing students to the joy of raising pigs. We have about 50% of our students who've never been on a pig farm, let alone held a pig. So we get in the pens, we hold pigs, we move pigs, and we sometimes laugh a little at some of the conversations that happen. But in addition to that, of course, we do work on the sow industry and research in our group. So where I have been over the last 15 to 18 years of my life has been focused on sow reproduction, sow longevity, and sow nutrition. Sows are, of course, something I'm very passionate about, and I believe they're the key to our success as an industry as we go forward. But where I feel like we've lacked over the last 15 years in terms of research is really how do we prepare that sow before we ever breed her the first time. We spent a lot of time talking about how do we feed this sow in lactation. Even a lot more recently, we've talked about gestation and different types of lysine diets. But when it comes down to it, we pretty much expect that sow to be ready to go as soon as she enters our barns and we breed her as a gilt. And I don't think that's fair to her. I think we're asking a lot of that animal if we're not setting her up correctly. And as some of our work alludes to, there's definitely differences between gilts and sows. And the question is what can we do to bridge that gap? I think we all recognize that as gilts that produce their first litters, those first litter offspring do not perform as well. In the grow-finish barns, they don't grow as well, their mortality is higher. And so certainly we want to start to understand what we can do to make them perform similar to offspring from sows. And so we'll talk a little bit about that research today. The first thing we're going to actually talk about, though, really goes into gilt development. And one of the challenges that we see in the industry beyond prolapses today is lameness. We have talked and talked for years about lameness. And I gave a talk 2 years ago in Germany, and I pulled up data 15 years apart with the common problems in the sow industry and what the primary reason for culling is, and it's lameness. It's not changed. We talk about it and we talk about it, but what have we really done to change it? And I'm not criticizing the industry because we've done a lot of wonderful things. We've started implementing organic minerals, and trace minerals have improved hoof health and they improve stability of the joints. But again, I think we still have more work that we can do in this field. So this is what we'll talk about today. First, we'll talk about joint health. Then we'll talk about vitamin and mineral status. And we'll talk not just in terms of what we see in the SAL facilities, but we'll also talk about what we are seeing currently in the nursery and the finishers. And then we'll talk a little bit about mammary gland development. Well, actually, we won't talk about that today. That is some of the work that we are working on that will be out probably in the next year. And that focuses, again, on gilt development and how we can alter mammary gland development early on. Okay, so today let's first talk about lameness. So again, lameness is nothing new to the industry. Certainly, we continue to monitor it and watch it, and we recognize that it's a large reason for culling in our populations. So why don't we do more of this work? Let's be honest, SAL research is expensive and it's timely. If I want to do a gilt development study and I want to follow that SAL for 3 parities, I'm looking at a 3 to 4 year study. It's a very long process, and so can be lengthy. Plus, again, it's hard to predict. We could have PERS breaks, we can have PED breaks, we can have lots of things that can interfere with how we interpret those results. And so we have been limited over the years when we've looked at why we have not continued to do more of this work. But yet when we look, sow mortality and removal rates are extremely high. In 870 farms over a 52-week period, the average death loss is 9%, and the removal rate is almost 50%. And again, these are not new numbers to us. These are numbers that we've seen for a while now. But again, we need to keep thinking about how we're going to respond to lower those numbers. So the work I'm going to talk about with lameness is work that's currently being done in our lab by Spencer Becker. She is our PhD student within the lab, and she presented some of this work at Midwest Animal Science this year. So I wanted to bring this back up, give everybody a chance who didn't get a chance to see it to see kind of what we're thinking and what we're doing. So our focus has really been on essential fatty acids and trying to understand the ratios of linoleic and linolenic acid. Most of us can recognize that even in the human side, there's a lot of good data that would suggest that linoleic and linolenic acid ratios at lower levels can improve joint health over time. And so, in fact, in pigs, we see the same thing. If we lower the N6 to N3 ratio and we supplement with more of the polyunsaturated fatty acids, it has been shown to mediate immune stimulation and reduce inflammation in the joint. And across all the species, we can recognize that high-fat diets can actually induce systemic inflammation and increase incidence of disease. So let's think about sows for a minute. When it's hot in the summer, we can feed an enormous amount of fat. And there are certain parts of the United States where it's hot year-round. They're feeding high fat levels throughout the year. And so there is the potential that we could almost be creating more systemic inflammation depending on the type of fat and the fatty acid ratios that we're feeding. And so this first study was really designed to understand if low-fat or high-fat diets change the amount of inflammation we see in the joint. Plus, we looked at 2 different linolenic to linoleic acid ratios. So in here, we looked at ratios of 12 to 1 and 23 to 1. So let me set this up a little bit. If we look at a current sow lactation diet today, and even in the GDUs, we're looking at about a 20% distiller's ration with about 2% fat. That's going to give us the 23 to 1 ratio. Okay, so what we're looking at here is changing that linoleic to linoleic ratio down to a 12 to 1 from the conventional 23 to 1. So this is really a 2-factorial process. We either have low fat or we have high fat, and then we have these 2 different ratios here. So what we have found is that when we feed these diets, we have an energy response, right? More fat we give the pigs, the better the growth rate. That would be something we would expect. Feed efficiency is improved in these studies as well. What we didn't quite expect in this study was that what we do see is, in fact, our gilts, when fed a 23-to-1 ratio, do not perform as well as the barrows, which that would be typical. But the 12-to-1 ratio, if you look there, the 12-to-1 ratio grows at the same rate as the barrows. A little interesting. Spencer and I scratched our head for many hours. That's not the primary objective of the study, so that's something we'll come back to later. But we did want to look again at inflammation. And so what we did, or what Spencer did during the study, was she went in and she took fluid from the joints, both at the beginning of the study and at the end of the study, as well as took blood samples throughout the study to try to understand inflammation and also look specifically at the joints and the inflammation there. She also exposed these animals to multiple rounds of vaccinations, much like we would expect to see in a gilt developer. Okay, so we tried to mimic very much what we're doing in the gilt developers today. And what we saw, or what she saw over time, is C-reactive protein increased for a period of time, and then it decreased as the animal matured. But TNF-alpha stayed pretty low, right? It went down and it stayed fairly low. We did not see any differences here in terms of linoleic to linolenic acid ratios. Okay, so nothing here would really indicate that either the low-fat or the high-fat diets or the ratios were changing TNF-alpha or C-reactive protein in the blood. Okay, so nothing would say that we fixed inflammation. Here she looks at it in the synovial fluid, and I apologize, the title came down onto the graph. We don't, again, see anything significant in terms of the ratio. We see some tendencies for energy to maybe alter the amount of C-reactive protein and TNF-alpha in the synovial fluid. But again, we're not picking up anything that's highly significant. Spencer also looked at a series of other enzymes within the body to try to understand how much activity we have going on. And we do see some differences between the high energy and the low energy diet and some of our liver enzymes. And again, that would be expected based on fat and how fat is processed. But we don't see anything else in terms of the ratio. So what we concluded with this study was that high energy and low energy diets don't really appear to do much in terms of changing inflammation. There is an indication that maybe there's something going on in the synovial fluid, but nothing that's significant. Spencer took time and she opened up all of the joints and looked for lesions within the joints as well, and did not really see anything in those joints. So from there, we really wanted to understand more about the ratio. Okay, so we felt pretty confident that it didn't matter if we were doing high fat or low fat diets. We really wanted to look at this ratio. And the ratio we're focused on is the N6 to N3, so linoleic to linolenic acid ratio. And what has been shown is that this high ratio can promote pathogenesis of many diseases, the inflammation, and depression and bone formation. Again, so high ratios, the 23-to-1, the 40-to-1s, the higher ratios are the ones we're concerned about. And we see a lot, particularly in pet food diets and so forth, where they're feeding a 4-to-1 ratio. Okay, 4-to-1's been, of course, promoted more for joint health. So Spencer set this study up where she looked at 4-to-1, 12-to-1, and 20-to-1 for those ratios. And she also took some time to look at some of the work that David Rosero has done on looking at different levels of linolenic acid in feed. Okay, so David's work, if you're not familiar with it, in sows would say that 100 grams of linolenic acid intake every day improves performance of those sows. So the question was, is that the same for gilts? Or is it not? Okay, so again, just to set this up, we looked both at performance, and then Spencer looked at the same parameters that we looked at in the first study. We don't see anything different there in terms of body weight at the beginning. But again, what we do notice is that we do see this similar pattern with the Barrows and the Gilts, where we have one treatment where the Gilts perform at a similar rate as the Barrows. It's at a different ratio than the last study, but it is there. Okay, but when we start looking at inflammation, we don't see a response. There is a significant difference there in linoleic acid, but we don't see anything in terms of the ratio. So let me clarify that. We don't see a difference in the ratios. So a 4-to-1 versus a 20-to-1 is not changing how much C-reactive protein or measurement of inflammation in the blood. And again, if we look at the synovial fluid, we don't see any difference with the ratios. So when we finish this study, we're still left with trying to understand why the Gilts are performing at a similar level as the Barrows with certain diets. But we're not seeing that changing the ratio is going to change potential influence of the joint health of the animal if we're feeding it in the Gilt development. Okay, so we're going to continue to work on this and try to understand what's going on, because there are some significant points in here and there are some trends, but we don't have all the pieces of information. So Spencer's working on her last study, and she's actually taking some females beyond a 285-pound weight. She's doing some heat checking now, and she's trying to look at performance reproductively. Because the large question we have is, if these Gilts grow at the same rate as a Barrow, are they reproductively sound? So that is what she's currently working on. So we're going to change gears a little bit, and we're going to talk about vitamin status. In the industry today, I think we've had a lot of questions come up about vitamin status. I've heard a lot of talk in the last 2 years about vitamin A in particular. But we've also, as we know in the past, have talked about vitamin D a lot. And when we talk to the veterinary diagnostic lab here at Iowa State, the references that they're using are back from 1994. So Poles et al., 1994 is where they're pulling their references. And if you look at that book, Sarah will tell you that there's a lot of interesting things about that book, but it is older data, right? That information is compiled and it's older. And so when we're looking at that, we're referencing values that are about 20 years old. And we're referencing values, particularly with vitamin D, where the pigs were housed outdoors. And vitamin D is elevated through the sunlight. And so as a nutritionist, or even as a veterinarian, when you receive that diagnostic report, how do you interpret that for today's pig? And how much has today's pig changed based on the housing methods that we're using? The other thing that our group has discussed quite a bit is where are the vitamins and minerals stored? It's very easy for us to go out and take a blood sample on a pig. But does that mean that's the right place to take that to measure that specific vitamin or that specific mineral? So our group set out to understand where we were at. And this is really just a pilot study, if you will. It was sponsored by the Iowa Pork Producers Association, where we took approximately 10 pigs from each phase of production. So we looked at suckling pigs, nursery, finishing, and sows. And we measured serum, diaphragm, and liver. Okay, so we chose diaphragm just because it was easy to identify and take out as representative of our muscle. So to walk you through where we're at, what you're seeing here, these are liver results. So you're going to see the vitamins and minerals that we tested there on your left, and then you'll see suckling pig, nursery finisher, and lactating sow. Now we did these during COVID, so we weren't able to get boars, and made for some interesting collection processes when we were locked down. But we were able to sample from a couple of different states to try to get an idea of what we're seeing. So these are just means. We have a full set of actual ranges as well, from what we saw from the low to the high. We didn't want to publish that in here today just because it's a lot of numbers on a slide. But what you do notice, and particularly when we look at vitamins that are stored in the liver, like vitamin A, we see that our sow has the highest level of vitamin A, and our nursery pig has the lowest. References for this, if we look at POLS data, for example, POLS data would tell you that a nursery pig, that a finishing pig today, or a finishing pig in their paper would have had a reference of about 200 to 400. Okay, so our vitamin A levels in a finishing pig are lower today than what's referenced in POLS. The lactating sow also ranges of about 200 to 400 in the POLS paper. So the lactating sow is very similar. Okay, so we do see some differences. We don't have vitamin D on here because we only did that one in serum, and we'll talk about that in a little bit. But we are seeing a few small differences. Does it mean that our pigs are deficient today? No. It does not mean that our pigs are deficient in vitamin A. Again, you have to think about how your pigs are housed. If your pigs were housed outdoors and they were eating grass, they were probably consuming carotenoids outside, and they probably had an elevated vitamin A level compared to an animal getting a corn soy-based diet today. But it is something we want to continue to monitor and watch. Okay, diaphragm, if we look at muscle, again, we'll have these values available in the paper, and we'll have them compared to what we're seeing with POLS. But we're seeing very similar levels in terms of what's present in the muscle as what's being published previously. The exception to that, I would say, is probably selenium, because we have switched to an organic selenium, and we tend to carry a little bit more selenium in the tissue when we use organic selenium. Okay, so that's just something to keep in mind. And then lastly is serum. Okay, so again, our numbers are changing a little bit. Serum levels for vitamin A, for example, have not changed from POLS to now. They're very similar. So liver is different, but serum is not. And again, when we think about vitamin A, it's stored in the liver. It's not stored in the blood. It's not a circulatory vitamin. And so does that mean that the blood is the best place to sample for vitamin A? The one I want to draw your attention to is vitamin D3. So our suckling pig today is testing at 3.95 nanograms per mil. Floor's work from many years ago would have suggested that the value was closer to 4.75, upwards to 9. I would say when we all saw the vitamin D concern many years ago, when we pulled suckling pig samples, we were between the 9 to 10 range. So we do see that the numbers have gone down again, but what does that mean? Right, so again, this is just an idea. It's just something for us to start thinking about. Are these concerns, or is this just something that's changed because we're putting pigs in barns and we're changing the way we feed them? We might want to think about that. Particularly, this is one I'm watching with vitamin D because of our concerns with lameness in the finishers and some of those challenges. But I'm not saying today that we have a deficiency issue. I'm just telling you this is what we're seeing today and how it compares to what we've seen in the past. So this was just a couple of examples I pulled just to kind of help you understand the differences if you're going to sample. Liver is in red, diaphragm is in black, and the serum is in blue. And so as you can see, if I'm going to sample an animal and I pull muscle, vitamin A really isn't going to be there, right? It's only going to be present in the sow tissue, so it probably isn't going to be the best place to assess vitamin A status. Liver here is going to probably give us the best estimate, and serum is going to be relatively low and relatively flat. Vitamin E, on the other hand, it is stored in the liver as well, but we also can see quite a few differences in serum if we look at the different phases of production. Okay, again, because vitamin E isn't stored in the muscle, that's probably not a place that you would want to take it from a diagnostic perspective. So hopefully this gives you just a little bit of an idea that we're starting to see some changes, or that we have seen changes since the published values from POLS. And again, to remind you that not every sample is going to give us the information we're looking for, particularly when we start talking about vitamins and minerals. The last one I pulled was copper, and again, copper is stored in the liver. And so we can see how that changes between the liver, the muscle, and the blood. Okay, so we're going to jump over to one more path, and I know it feels like we're jumping a lot. Our lab does change paths quite a bit because we like to do things that interest the students, and so a lot of times I'm along for the ride. Which makes it fun because, as I told them, my goal is to help you, and as long as we stay with SOWs and try to account for that, I'm pretty happy. So the last one that I'll share with you today that our group has been working on is involving the enteromammary pathway. And the enteromammary pathway is something that's been talked about for years in humans, but not really talked about in pigs. And when I started searching about 2 years ago, I couldn't find a paper on it in pigs. So the question was, does the pathway exist? And what is this pathway? So what this pathway is, it's a pathway where immune cells will travel to the intestine of the animal. It will grab bacteria or viruses, not kill them, right? Immune cells should kill things, but they don't. They don't kill them. They carry those bacteria and viruses up through the bloodstream and deposit them into the mammary gland, where the piglet will then receive it in the milk. Okay, so that's how we get the establishment of a fecal microbiome. That's how we get our microbiome in newborns, whether it's a new pig, a new person. It's happening through that process. Okay, so why is this important? So this is based off a theory of mine that we're still working on, and I don't have proof today yet. But in humans, organisms like HIV, cytomegalovirus will go through this process. And when I was in production, we struggled with rotavirus C for many, many years. And we would have pigs within 6 hours of birth be very symptomatic with rotavirus. Have lots of scours, get very sick, but yet we knew the rotavirus wasn't passing through the placenta. So how could the pigs be getting so sick so quickly? And so the question that I have is, are we in fact passing bad organisms as well as the good things through this pathway? So the students and I sat out. We collected a large number of sow milk samples, colostrum samples, and blood samples, as well as fecal from sows as they were farrowing. And we looked at both gilts and sows, and we'll talk a little bit about differences here in a minute. And we just set out to understand what's present in the gut, what's present in the feces, what's present in colostrum, and what's present in blood. And I'm still learning all the organisms and what they all do, and so I don't expect this to ring much to many people in the room. But what we do see is that there are certain organisms that are predominant in things like colostrum. So you can see up there at the top, Clostridium sensu stricto 1. Okay, we all know what Clostridium is, right? So Clostridium sensu stricto 1, it is present in colostrum, but it is not the same Clostridium that we would associate with disease in pigs. Okay, this actually is an interesting organism because we have found that in poultry, for example, certain levels of this organism can cause necrotic enteritis or regulate necrotic enteritis. Okay, so if we have a large amount of this in the colostrum, we would think then it's potentially regulating some of the harmful pathogens that our piglets could be exposed to. The other one that I thought was interesting was the next one below that, which is mycoplasma. Mycoplasma was found on the highest level in the blood. Wouldn't surprise you then if I told you that this herd had just recently gone through a mycoplasma exposure, right? So it kind of made us feel good that, okay, maybe the data makes sense because we found mycoplasma when we knew there was mycoplasma present in the herd. And then the last one there, or the third one on the list, is lactobacillus, which is found mainly in the feces. And again, when we think of lactobacillus, we think of that as an organism that we see in the gut, and it's supposed to be a good organism. Okay, so we are seeing differences there. We were also looking to see what was common across all three. And we were able to find a few organisms that are common across all three types. So it does indicate that, or it does suggest that the pathway is there. But what's more interesting to us right now that Dalton and I are working on is trying to go through parity differences. Okay, remember how I told you at the beginning of the talk that we're trying to figure out how we can help the gilt offspring perform similar to sow offspring. And this is an example of what we're seeing is there are in fact differences between the gilts and the sows when we start looking at some of the organisms that are present. And certainly in the different types. So for example here, because mycoplasma was mostly present in the blood, we do see that there are significant differences in the blood between the gilt and the sow. And that is further expressed over in the colostrum as well as in the feces. So we do know that in this facility, which we would expect, the gilts are shedding more mycoplasma than the sows. Okay, so I don't think this one would be anything new, but other organisms that we're looking at would be things like that Clostridium sensostricta 1. That is also different between gilts and sows. Okay, so if I have something that's different, particularly when we talk about something that can protect the gut, there could be some indication there that we could make some changes. Okay, so as I wrap up, that's just kind of give you a short glimpse into what our group is working on. Other projects that they've been busy working on, as I mentioned, Chloe's been working on some compensatory growth and feed space allowances and finishing. Mainly understanding in the floor space allowance what we can do nutritionally to see if we can override the restriction that we see as we restrict growth, as we restrict floor space. We're also looking, Sarah's spending some time looking at proper handling of samples and sampling collection methods for those vitamin and mineral analysis. So as our data shows, there are different tissues that we want to handle to look at the different vitamins and minerals, but how are we processing it? Right? Some of our vitamins are light sensitive, so is it okay to leave the blood tube sitting out while I'm collecting other things? Should they be cold? How long can they sit out? Those types of questions. And then lastly, Dalton's been working on some environmental stewardship projects where he's been looking at odor and trying to understand ways to reduce odor. So with that, I'm leaving plenty of time for questions. I do want to thank the graduate student team, Spencer Becker, Sarah Elfson, Chloe Hagan, and Dalton Humphrey. They put in a lot of work on these projects, and I think they have a little bit of fun in the process. And I do want to acknowledge those who have supported the research, particularly National Pork Board, Iowa Pork Producers Association, and Iowa Select, as well as the folks in the vet school for helping us with the vitamin work, and Dr. Schmitz-Esser for helping us with the microbiome interpretation. So with that, I'm going to leave plenty of time, as I mentioned, for questions. Are there any questions? Yeah.
Laura, on your work where you were looking at the pro-inflammatory and the varying your omega acid ratios, is there any thought that looking at anti-inflammatory cytokines like IL-10 and some of those others might have helped further define the response? And then secondly, along with those lines, I don't know the answer to this. It's just a thought out there. But Dr. Levin up here at the USDA has done a lot of work on looking at what she phrases as training the immune system.
Mm-hmm.
And I'm just curious that with the work that you've done here, is there any potential that the question of the high fat or high energy diet, is it a dose by duration kind of a thing where looking at growing barrows and gilts versus an older mature sow that may have had a couple of parities under her belt might get a different response? I don't know. I'm just thinking out loud.
Mm-hmm. Yeah, I think you're asking some good questions. So IL-10 is, Spencer's still working on some lab work, and some of those cytokines are still what she's working on. So they are in the process of being reviewed. In terms of immune cell training and age, I don't disagree. I think there is some advantage on age and immune cell training. I still challenge the concept, though, is what can we do to help push that gilt farther? Right? As an industry, if we're talking that a third of our pigs never make it to market, what are we doing to try to change that? And there may not be, right? This may be a biological thing where we're stopped, right? The body doesn't allow us to overproduce cytokines or we die type of situation, or it might not be. And so, I think that's really kind of where we're at, is just trying to see what can we do to recognize the challenges and see if we can push it, and we may not, right? The sow may always inherently be more immune just because she's seen a lot more, just like us, right? Unfortunately, age brings,
Age and vaccination.
right. Any other questions? All right. Well, thank you all for your time.